Welcome to the fascinating world of microbial metabolism. Microorganisms, though tiny, play a massive role in the ecosystem by driving essential biochemical reactions. Their metabolic activities are responsible for processes ranging from nutrient cycling to energy production. In this video, we'll explore how microbes obtain energy, break down nutrients, and contribute to life on Earth. Whether you're a student, a science enthusiast, or just curious, this journey will help you appreciate the invisible engines of our biosphere. Let's begin by understanding what metabolism means in the context of microbes and why it's crucial for their survival and for the planet as a whole. Metabolism refers to all the chemical reactions that occur within a living organism to maintain life. For microbes, metabolism is the sum of all processes that allow them to grow, reproduce, and respond to their environment. These processes can be divided into two main categories: catabolism, which breaks down molecules to release energy, and anabolism, which uses energy to build cellular components. Understanding these two branches is key to grasping how microbes thrive in diverse environments, from deep oceans to our own bodies. Metabolism is the process by which microbes break down complex molecules into simpler ones, releasing energy in the process. This energy is then used to fuel other cellular activities. For example, bacteria can break down sugars, fats, or proteins depending on their environment and genetic makeup. The energy released during catabolism is often stored in molecules like ATP, which acts as the cell's energy currency. Catabolic pathways are essential for microbial survival, especially in nutrient-limited environments. The flip side, anabolism, is all about building. Microbes use the energy derived from catabolism to construct complex molecules from simpler ones. These molecules include proteins, nucleic acids, and cell wall components, all vital for growth and reproduction. Anabolic reactions are crucial for repairing cellular damage and for the synthesis of new cellular structures. Without anabolism, microbes couldn't multiply or adapt to changing environment. Adenosine triphosphate, or ATP, is often called the energy currency of the cell. Microbes generate ATP through various metabolic pathways, and then use it to power essential functions like movement, transport, and biosynthesis. The production and consumption of ATP is a constant cycle, ensuring that the cell always has the energy it needs. Understanding ATP's role helps us see how energy flows through microbial system. Enzymes are proteins that speed up chemical reactions in microbial metabolism. Without enzymes, most metabolic reactions would occur too slowly to sustain life. Each enzyme is specific to a particular reaction, ensuring efficiency and regulation. Microbes produce a wide variety of enzymes, allowing them to adapt to different environments and utilize diverse nutrients. Enzyme activity is tightly controlled, reflecting the cell's needs and environmental condition. Metabolic pathways are a series of interconnected chemical reactions. In microbes, these pathways can be linear, branched, or even circular. Each step is catalyzed by a specific enzyme, and the product of one reaction becomes the substrate for the next. This organization allows for efficient regulation and adaptation. Examples include glycolysis, the citric acid cycle, and fermentation. Understanding these pathways gives insight into how microbes manage energy and resources. Glycolysis is one of the most fundamental metabolic pathways in microbes. It involves the breakdown of glucose, a simple sugar, into pyruvate, generating ATP and NADH in the process. This pathway is anaerobic, meaning it doesn't require oxygen, making it especially important for microbes living in oxygen-poor environments. Glycolysis is the starting point for many other metabolic processes. Fermentation is a metabolic process that allows microbes to generate energy in the absence of oxygen. After glycolysis, pyruvate can be converted into various end products like lactic acid, ethanol, or gases. Fermentation is crucial for many microbes, especially those in environments where oxygen is scarce. It's also the basis for food production processes like yogurt, bread, and alcohol. When oxygen is available, many microbes use aerobic respiration to extract the maximum amount of energy from nutrients. This process involves the citric acid cycle and the electron transport chain, resulting in the production of much more ATP compared to fermentation. Aerobic respiration is highly efficient and allows microbes to thrive in oxygen-rich environments. Citric acid cycle, also known as the Krebs cycle, is a central part of aerobic respiration. It takes the products of glycolysis and further breaks them down, releasing carbon dioxide and transferring energy to molecules like NADH and FADH2. These molecules then feed into the electron transport chain, driving ATP production. The citric acid cycle is a hub for many metabolic pathways in microbes. Electron transport chain is the final stage of aerobic respiration. Here, electrons from NADH and FADH2 are passed through a series of proteins embedded in the cell membrane. This transfer of electrons creates a flow of protons, generating a gradient that drives the synthesis of ATP. Oxygen acts as the final electron acceptor, forming water. This process is the main source of ATP in aerobic microbes. Microbes can respire without oxygen by using alternative electron acceptors like nitrate, sulfate, or carbon dioxide. This process, called anaerobic respiration, is less efficient than aerobic respiration but allows microbes to survive in oxygen-depleted environments. Anaerobic respiration is important in natural cycles, such as nitrogen and sulfur cycling, and in wastewater treatment. Not all microbes rely on breaking down organic molecules for energy. Some, like cyanobacteria, can capture light energy through photosynthesis. These microbes use sunlight to convert carbon dioxide and water into glucose and oxygen. Microbial photosynthesis is vital for producing oxygen and supporting food webs, especially in aquatic environments. Microbes, known as chemolithotrophs, obtain energy by oxidizing inorganic compounds like ammonia, hydrogen sulfide, or iron. This unique metabolism allows them to thrive in extreme environments, such as deep-sea vents or acidic hot springs. Chemolithotrophy plays a key role in nutrient cycling and supports ecosystems where sunlight is absent. Microbes can be classified based on how they obtain carbon. Heterotrophs rely on organic compounds from their environment, while autotrophs can fix carbon dioxide to build their own organic molecules. This distinction shapes their ecological roles and determines where they can live. Both types are essential for maintaining balance in ecosystems. Earth microbes have the remarkable ability to convert atmospheric nitrogen into forms usable by plants and other organisms. This process, called nitrogen fixation, is essential for agriculture and natural ecosystems. Nitrogen-fixing bacteria often form symbiotic relationships with plants, providing them with vital nutrients in exchange for shelter and food. Microbes specialize in metabolizing sulfur or iron compounds. These metabolic processes are crucial in environments like hot springs, deep-sea vents, and mining sites. Sulfur and iron metabolism contribute to nutrient cycling and can influence the chemistry of entire ecosystems. These microbes often play a role in bioremediation and industrial processes. Methanogens are a group of archaea that produce methane as a byproduct of their metabolism. They thrive in anaerobic environments like wetlands, digestive tracts, and landfills. Methanogenesis is a key step in the carbon cycle and has implications for greenhouse gas emissions and renewable energy production. Microbes are not just found in the environment, they also inhabit our bodies. The human microbiome is a complex community of microbes that help digest food, produce vitamins, and protect against pathogens. Their metabolic activities are essential for our health and well-being, influencing everything from immunity to mood. Microbes rarely exist in isolation. Instead, they form complex communities where different species cooperate and compete. These interactions can enhance metabolic capabilities, allowing communities to break down complex substances or survive harsh conditions. Studying microbial communities helps us understand processes like soil fertility and wastewater treatment. Biofilms are structured communities of microbes attached to surfaces. Within biofilms, microbes communicate and share resources, creating a protective environment. Biofilms can form on natural surfaces, medical devices, and industrial equipment. Their metabolic cooperation makes them resilient and sometimes challenging to control. Microbes use chemical signals to communicate with each other, a process known as quorum sensing. This allows them to coordinate activities like biofilm formation, virulence, and nutrient acquisition. Quorum sensing is a fascinating example of how even single-celled organisms can behave collectively, adapting to changing conditions. Microbial metabolism has a profound impact on the environment. Microbes drive nutrient cycles, decompose organic matter, and influence soil and water quality. Their metabolic activities can also help clean up pollutants through bioremediation. Understanding these processes is vital for environmental management and sustainability. Microbial metabolism is harnessed in many industries. From producing antibiotics and enzymes to fermenting foods and treating waste, microbes are invaluable. Their metabolic diversity allows for the development of new biotechnologies, making processes more efficient and sustainable. The future of industry is closely tied to our understanding of microbial metabolism. Certain microbes can break down pollutants and toxic compounds, making them essential for cleaning up contaminated environments. This process, known as bioremediation, uses microbial metabolism to transform harmful substances into harmless ones. Bioremediation is used in oil spill cleanup, wastewater treatment, and soil restoration. Any antibiotics are natural products of microbial metabolism. Microbes produce these compounds to compete with other organisms in their environment. By studying and harnessing these metabolic pathways, scientists have developed life-saving medicines. The ongoing search for new antibiotics relies on exploring microbial diversity and metabolism. Microbial fermentation is the foundation of many traditional and modern foods. From bread and cheese to yogurt and beer, microbes transform raw ingredients into delicious products. Their metabolic activities create unique flavors, textures, and nutritional benefits. Fermentation is a testament to the power of microbial metabolism in our daily lives. Microbes can convert organic materials into biofuels like ethanol, methane, and hydrogen. These renewable fuels are produced through metabolic processes such as fermentation and anaerobic digestion. Biofuel production offers a sustainable alternative to fossil fuels and helps reduce greenhouse gas emissions. Advances in genetic engineering allow scientists to modify microbial metabolism for specific purposes. By inserting or deleting genes, microbes can be tailored to produce valuable compounds, degrade pollutants, or resist diseases. This technology is revolutionizing medicine, agriculture, and industry. Synthetic biology combines engineering and biology to design new metabolic pathways in microbes. This field enables the creation of organisms with novel functions, such as producing pharmaceuticals or bio-based materials. Synthetic biology holds promise for addressing global challenges in health, energy, and the environment. Microbes must carefully regulate their metabolic activities to survive and thrive. Regulation ensures that energy and resources are used efficiently and that the cell can respond to changes in its environment. Mechanisms include feedback inhibition, gene regulation, and signal transduction. These controls are vital for maintaining balance and adaptability. Microbes known as extremophiles have evolved unique metabolic strategies to survive in harsh conditions like high temperature, acidity, or salinity. Their enzymes and metabolic pathways are specially adapted, making them valuable for industrial applications and scientific research. Studying extremophiles expands our understanding of life's possibilities. Microbial metabolism has evolved over billions of years, shaping the diversity and adaptability of life. Horizontal gene transfer, mutation, and selection drive the evolution of new metabolic capabilities. This evolutionary flexibility allows microbes to colonize nearly every environment on Earth. Microbial metabolism influences global climate by affecting greenhouse gas levels. Processes like methanogenesis and denitrification release methane and nitrous oxide, potent greenhouse gases. Conversely, microbes also help sequester carbon and mitigate climate change. Understanding these processes is crucial for environmental policy and climate action. Microbes often form symbiotic relationships with plants, animals, and other microbes. These partnerships can enhance metabolic capabilities, provide nutrients, or protect against disease. Symbiosis is a driving force in evolution and ecosystem function, highlighting the interconnectedness of life. Some microbes cause disease by exploiting metabolic pathways in their hosts. Understanding microbial metabolism helps us develop strategies to prevent and treat infections. Targeting metabolic processes is a promising approach in the fight against antibiotic resistance and emerging pathogens. Metagenomics is the study of genetic material recovered directly from environmental samples. This approach allows scientists to explore the metabolic potential of entire microbial communities, revealing new enzymes, pathways, and organisms. Metagenomics is revolutionizing our understanding of microbial diversity and function. Modern omics technologies such as genomics, proteomics, and metabolomics provide detailed insights into microbial metabolism. These tools help identify genes, proteins, and metabolites involved in metabolic pathways. Integrating omics data accelerates discoveries and applications in medicine, agriculture, and industry. Study of microbial metabolism is rapidly advancing. New technologies and interdisciplinary research are uncovering novel pathways and applications. From sustainable energy to personalized medicine, the future holds exciting possibilities. Continued exploration will deepen our understanding and unlock the full potential of microbes. Despite progress, many challenges remain in studying microbial metabolism. Complexity, diversity, and the difficulty of culturing many microbes limit our knowledge. Advances in computational biology and high-throughput techniques are helping to overcome these barriers, paving the way for new discoveries. Biotechnology relies heavily on microbial metabolism. Engineered microbes are used to produce pharmaceuticals, biofuels, and specialty chemicals. Their metabolic versatility makes them ideal platforms for innovation. As our understanding grows, so does the potential for new biotechnological solutions. With advances in microbial engineering and synthetic biology come ethical questions. How should we balance innovation with safety and environmental responsibility? Ongoing dialogue among scientists, policymakers, and the public is essential to ensure responsible development and use of microbial technologies. Raising awareness about microbial metabolism is crucial for public health, environmental stewardship, and scientific literacy. Educational initiatives and outreach programs help people appreciate the importance of microbes and their metabolic activities. An informed public can better support research and innovation. Microbial metabolism is a cornerstone of life on Earth. From sustaining ecosystems to driving technological innovation, microbes and their metabolic processes touch every aspect of our world. As we continue to explore and harness their potential, we unlock new opportunities for health, sustainability, and discovery. Thank you for joining us on this journey into the invisible yet powerful world of microbial metabolism.